Good morning, ladies and gentlemen. I'm delighted to be in Carlsbad this morning and to introduce you to Avectas and the work we're doing to develop a next-generation cell engineering platform. So I've been reflecting on what I've been hearing at this conference over the last 2 to 3 days, and what occurs to me is that this industry is at a point where the rubber is hitting the road, if you like, in terms of the complexity and the cost of the therapies which have been developed. And the participants in this industry are all looking at addressing that cost and complexity through their own paradigm, whether it's logistics, biology, immunology. And I'm trying to connect those thoughts with what we're doing at Avectas to address cost and complexity in intracellular delivery. And our platform, Solupor, introduces simplicity and effectiveness, and I'd like to spend the next few minutes showing you how that's affected. So we can see the very familiar adoptive therapy paradigm here. And currently, developers are introducing a very wide array of cargoes into cells, including DNAs, mRNAs, gene editing tools. And these, in the first generation of advanced therapeutic products, were done one at a time, if you like. However, the territory that we're moving into now is going to require much more sophisticated interactions where we're delivering multiple entities into cells, multiple modifications for sophisticated products. This addresses two key factors that the industry has to address. One of those is allogenicity, whereas the approvals to date are autologous. Allogeneic needs to be embraced to address cost and complexity. And the second one is solid mass tumours. So we can see in this pie chart that although the product approvals to date are wonderful, they're in the blood malignancies, and we can see the slice of that pie is small relative to the overall tumour burden. And to address these solid mass tumours is going to require sophisticated approaches. It's also going to require cell engineering that produces cells which are fit to go into the body and do their job. And we see on the right-hand side here an engineered CAR T cell going into the body, into a solid mass tumour. It's a hostile place to go for a cell. So that cell needs to be as fit and as functional post-engineering going into that environment as possible. So the key challenge, if the catch cry of the relator is location, 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 the drug developer or therapeutic developer, it's delivery, delivery, delivery. And delivery is the key challenge. Intracellular delivery is the key challenge in this business, to deliver complex molecules into cells in a way that leaves the cell functionally fit post-engineering. And whilst viral transduction has and will continue to play a key role in that, the cost and complexity, which are well understood in accessing viruses, manufacturing them and so forth, are causing developers to look to non-viral methods. And key amongst those non-viral methods, the go-to, if you like, is electroporation. And over this discussion, we've done some comparison work against electroporation of our new cell engineering platform, Solupor. So our solution, this is what it looks like. It is a combination of a device and a reagent. The device you're looking at is there's no scale there, so it's about the size of an American cereal bowl. It engineers about 100 million cells, which is a choice which is suitable for gene editing. We can expand that to a log greater as necessary. So it's the right number of cells, and it is being developed as a closed, automated, GMP-aligned platform. So it works very simply. We introduce cells in suspension into the device. They are filtered through a filter at the base of the device, and they're disposed on an engineered filter. We then spray the device with a chemically defined vehicle containing a permeabilization agent and the cargo of choice. There's a brief incubation, and then we put a stop solution on, and we simply resuspend the cells. The resulting cells tolerate the process extremely well, they're highly functional, and we go on to show this in several ways. So, first of all, in demonstrating some data on the basic performance of the system, we delivered a number of model cargoes, which you would expect in this industry. So, messenger RNA into human primary T cells, and showing in this data different activation forms. The delivery is highly efficient for these purposes. We then show the versatility of the platform, and we show on the left that we synthesized our own CD19 CAR cells, and on the right-hand side, embracing the request or interest in delivering DNA, we show delivery of linear DNA here. The interesting aspect of this data is the high viability of DNA, which is of cells which have DNA soluparated into them. This is in contrast to electroporation, which exhibits a lot of toxicity. We then go on to demonstrate a number of different gene editing platforms, and we get excellent editing efficiency in the types of locus that are expected in this industry, TCR alpha, PD1 knockdowns, and so forth, over a number of different platforms. So within human primary T cells, we can edit effectively with the soluporation platform. At this point, we draw a comparator in, so having delivered model compounds and done some gene editing, we want to then see how the platform compares with nucleofection, which is the go-to alternative. So a fantastic ready reckoner for the health and fitness of cells is the extent to which they proliferate subsequent to engineering. And we can see in the graph on the left here that cells which are engineered using soluporation proliferate the same as untreated cells. However, the nucleoaffected cells experience a lag in the culture. This is something widely reported and experienced. We can see on the right-hand panel we get the same results when we do this with freeze-thaw as well. We then went to look at a subcellular level of what's happening within the cell in subsequent to the engineering process. And we looked at a panel of 600 immune-related genes. We nucleoaffected and soluporated cells. And in this particular study, we did what we term a mock nucleoaffection and a mock soluporation. So in order to ascertain the performance of the platform rather than the cargo, we introduced a dead or no cargo at all. So we're just looking at the process. So in the top data here, the black part of the disk represents genes which have been upregulated or downregulated as a result of nucleoaffection. And we can see that approaching half of the genes have been at the 6-hour point, which is critical in how that culture takes off, have been modified. By 24 hours, that picture has settled down a little bit, but you still have a significant number of genes modified. In comparison, the soluporated cells exhibit very little evidence of up or down regulation of genes, both at the 6-hour and the 24-hour time point. The data on the right is the same data presented as a heat map, and it makes for an impressive picture, if you like. Quite a bit of perturbation here relative to relatively little perturbation. So we went on then to look at how were those pathways which were up or down regulated indicated in the performance of the cells in the body. And we can see the usual suspects: activation, metabolism, exhaustion, and so forth. We can see in the nuclear-affected cells that these are significantly altered. We can see in the soluparated cells that these are not altered that much at all, which is a remarkable result. This adds to the story of being able to put these cells back into the body and have them perform as if they were unengineered. We went on to look at the cytokine profile of the cells, and notably again, the red line on these here represents nucleofaction, and we can see that cytokine levels, IL-2 levels and IL-8, were increased in the nucleoaffected, the electroporation of cells via nucleofaction. However, they are not different from untreated with, there's no such elevated levels with soluporation. We went on to engraft engineered cells into a mouse model, so it's a humanized mouse, and we see that the soluparated cells engraft very well, whereas the nucleoaffected cells do not engraft into the mouse in this particular model. So having developed the technology, we wanted to externally validate this, and we have won some valuable external partnerships in North America and in the United Kingdom. So we're working with therapeutic developers who are working in gene editing and in CAR-T type areas. They're evaluating our platform and using it to engineer cells for their therapeutic development. In addition to that, we're working with research institutions in the US and in the United Kingdom, where we're seeding our technology early on in the development process, such that our technology platform can be adopted early and then pulled through as those products are developed and come closer to the clinic. We've picked up some terrific citations, particularly Nature Biomedical Engineering and chemical reviews recently. And our work, I guess, has been endorsed through funding from the European Union at quite significant levels. We've a growing patent portfolio, which is managed in Boston by Mintz-Levin partners, bringing together the crystallizing and protecting this platform for us. So in summary, Soluporation, which we've been developing, is a next generation cell engineering platform, if you will, a 21st century cell engineering platform for 21st century cell therapy development. It's designed to be easily adoptable by therapeutic developers. It's closed, it's scaled, it's GMP aligned, both in terms of the device and the reagents which it uses. And critically, it's efficient and it's gentle on cells. These qualities enable this technology to reduce time and reduce the cost of manufacture by having higher yields, less cell toxicity, less cell death in the engineering process. So the platform lends itself to solving some of the problems that the industry as a whole is experiencing. And our objective with this platform is to partner in the development of new therapies with therapeutic developers like yourselves and people at this conference and beyond. So thank you very much indeed.